Today we are going to see about chromosomes. Introduction. The chromosomes are the thread-like structures which are present in the nucleus, which carries the genetic information from one generation to the other. The chromosomes play a vital role in the cell division, heredity, variation, mutation, repair, and regeneration. Each chromosome is made up of a tightly coiled DNA around the protein called the histones that supports its structure. The chromosome means the colored body that refers to its staining ability by some dyes. The term chromosome was first coined by W. Walter in 1888. The number of chromosomes in any species is constant for all the cells. The chromosome number varies in different species. For example, a nematode species contains only 2 chromosomes in a cell, whereas in a protozoan species contains as much as 1600 chromosomes in the cell. In human cells, it contains 23 pairs of chromosomes, of which 22 pairs are autosomes, and otherwise called as somatic chromosomes, and one pair of allosomes, it is otherwise called as sex chromosome. Karyotyping is the study of the structure of chromosomes which are present in a species. By this figure we can understand how the chromosomes are located and how it is made. A cell contains the nucleus. Inside the nucleus, the chromosomes are there. The chromosome is formed of a highly coiled DNA. Here the DNA is that the DNA is coiled around the histone particles, like beads. Histone particles. This is the protein molecules. And this is the double-stranded DNA. Next is the structure of chromosome. The main parts of the chromosomes are the chromatid. This is the chromatid. Here these 2 chromatids, that is the sister chromatids. And the chromonema. Chromomeres. And the centromeres. The centromere is located in the center. The secondary constriction, I will show it in the other figure, or it is otherwise called nuclear organizer. The telomeres, the terminal part is called the telomeres. And the satellite DNA. Then the chromatids. The chromatids are each chromosome has 2 symmetrical structures called the chromatids. There are the sister chromatids which are visible in the mitotic metaphase condition. At anaphase of mitotic division, the sister chromatids separate and migrate to the opposite directions, that is the opposite poles. Next is the centromere and the kinetochore. The centromere is the sister chromatids are joined by means of this centromere. During the cell division, the spindle fibers are attached at the centromere area. The number and position of the centromere differs in different chromosomes. The centromere is also called the primary constriction. In the centromere divides the chromosome into 2 sections or the 2 arms. The shorter arm is called the P arm and the longer arm is called the Q arm. It contains a disc-shaped kinetochore which has the specific DNA sequence with a specific protein bound to them. The kinetochore provides the center for the polymerization of tubulin proteins and the assembly of the microtubules. The secondary construction and the nuclear organisms. Besides having the primary construction, the chromosomes possesses the secondary construction at any point of the chromosome. And this figure will clearly show the secondary construction area. And this is the satellite DNA. This is a constant in their position. The secondary constructions are constant in their position as well as their extent. These constructions are useful in identifying the particular chromosome in a set. The secondary construction can be identified from the centromere at the anaphase condition because there is bending only at the centromere, that is the primary construction. The secondary construction, which contains genes to form the nucleoli, are known as the nuclear organizer. The telomere is the terminal part of the chromosome. These are the specialized ends of the chromosome which exhibit the physiological differentiation and the polarity. The telomeres are polar, which prevent the fusion of the chromosomal segments. If the chromosome breaks, the broken ends can fuse with each other due to the lack of the telomeres. Next is the satellite. The satellite we have seen in the last slide. We have seen the satellite DNA. The chromosomal region distal to the secondary construction, that is the region between the secondary construction and the nearest telomere, is known as the satellite. It is an elongated segment that is sometimes present on the chromosome at the secondary construction. The chromosomes with the satellite is also called a SAT chromosome. The chromatin is the chromosomes are visible as thin chromatin fibers which are present in the nucleoplasm at the interphase condition. During the cell division, these chromatin fibers contents and the chromosomes are visible with distinct features. The darkly stained condensed region of the chromatin is known as the heterochromatin. It contains a highly packed DNA, that is tightly packed DNA, which is genetically inactive. But the lightly stained diffused region of the chromatin is known as the euchromatin. It contains genetically active and loosely packed DNA. At the prophase condition, the chromosomal material is visible as thin filaments known as chromonemata. At the interphase, the bead-like structures are visible, which are an accumulation of the chromatin material called the chromomere. The chromatin with the chromomere looks like a necklace with beads. Coming to the types of chromosomes, first one is the autosomes and the sex chromosomes. For example, in human, the chromosomes are of two types, that is autosomes and the sex chromosomes. The autosomes are otherwise called the somatic chromosomes and the sex chromosomes are otherwise called the allosomes. The genetic traits that are linked to the sex of a person are passed on to the sex chromosomes, that is the allosomes, and the rest of the genetic material is present in the autosome, that is the somatic chromosome. If humans having 23 pairs of chromosomes in their cells, of which the 22 pairs of chromosomes are autosomes and one pair of chromosomes is the sex chromosomes, making a total of 46 chromosomes in each cell. On the basis of the number of centromeres, the chromosomes are further classified into monocentric, dicentric, polycentric, acentric, and holocentric. The monocentric chromosomes are having only one centromere, but the dicentric chromosomes having two centromeres. That is, during the chromosomal aberration, the chromosomes break and fuse together with the two centromeres. They are also unstable as the centromeres tends to migrate to the opposite poles, resulting in the fragmentation. Then the polycentric chromosomes are having more than two centromeres. Then acentric, that is absence of centromere. So these centromeres, chromosomes are having no centromeres. Such chromosomes represent freshly broken segment of the chromosome which do not survive for a long time. In the holocentric, chromosome, the centromeres are diffused or non-located with indistinct. They're diffused throughout the length of the chromosome. The basis of the location of the centromeres, the chromosomes are further classified into telocentric, acrocentric, metacentric, and submetacentric. The telocentric chromosomes are rod-shaped chromosomes with the centromere present at the proximal end, that is the terminal end, so that the chromosome has not just having only one arm. And the acrocentric chromosomes are also rod-shaped chromosomes with the centromeres occupying in a subterminal position. So one arm is very long and the other arm looks very short. The submetacentric chromosomes are looking like a L-shaped or J-shaped with the centromere slightly away from the midpoint, it giving rise to two unequal arms. Metacentric chromosomes. The metacentric chromosomes are V-shaped chromosome in which the centromere lies in the middle of the chromosome, giving rise to the two equal arms. Here you can see the different types of chromosome based on the position of the centromere, and this is the acrocentric, that is in acrocentric, it is located at the terminal position. The centromeres are located in the terminal position. One arm's looking very short and the other arm's looking long. In submetacentric, it is in the subterminal position. So one arm also short, but when compared to acrocentric, it is long, and the other arm is very long. In metacentric, it is located in the middle of the central chromosome. In telocentric, it is located at the terminal, that is the polar region. It is the proximal end of the chromosome. Functions of chromosomes. The main function of the chromosome is to carry the genetic material from one generation to the other generation. The chromosomes are of great importance in determination of the phylogeny and the taxonomy of the species, because the number of chromosomes is a constant for a particular species. The chromosome play an important role and act as a guiding force in the growth, reproduction, repair, and the regeneration process that is important for the survival because of having the genetic code. The chromosome protects the DNA from getting tangled and damaged. It also helps in the determination of sex, that is out of the 20, for example in human, out of the 23 pairs of chromosomes, one pair is sex chromosomes. So the females are having two X chromosomes and the males are having one X chromosome and one Y chromosome. So the sex of the baby is determined mainly by the chromosome passed from the male. Then the histone and non-histone proteins helps in the regulation of the gene expression. The spindle fibers attached in the centromeres helps in the movement of the chromosome during the cell division. Each chromosome contains thousands of genes that precisely code for multiple proteins present in the body. Thank you.